This is screen capture video unit 10.1 and it covers cellular division. If you're following along in the Pearson text Biology by Campbell et al., it would be chapter 8. If you go back in time and you consider a person by the name of Virchow in the 1800s, 1858, he supposedly came up with an idea that helped with the cell theory, and that was all cells come from pre-existing cells. And that's a tenet, or that's a rule that biologists work from today. That's a general understanding that if you see a cell, it has come from a pre-existing cell. Cells will reproduce or divide in order to allow for growth or repair or to replace what is lost. They'll also divide for asexual reproduction, and they'll divide for sexual reproduction, and we'll talk a little bit about each of those, especially the asexual reproduction and the sexual reproduction. With the asexual reproduction, we've talked a little bit about that in the past with plants, and that is where cells will divide and no sex cells are being produced, no gametes as they're called, sperm or ova. But just more cells are produced, and there's enough structure that can break off or be separated to form a new organism. We saw that with the stolons, with the strawberries. It was a very good example. With sexual reproduction, you actually have sexual cells produced, which are called gametes. That would be the spermatozoa and the oocyst, the ova. And those can come together at some point and fertilization can occur and a new individual can be produced. The cellular division that we're going to be talking about first is binary fission. And this is something that prokaryotic cells do. When you think about prokaryotic cells, think about bacteria. This is the way that bacteria reproduce. There's no sexual reproduction. It's all asexual reproduction for bacteria. They don't exchange genetic material as eukaryotic cells do. The closest they come to it is a process called conjugation. That's just one way, and that's where one bacterium will donate plasmid, small loop of DNA to another. But that still is not sexual reproduction. The binary fission involves the bacterium replicating by DNA replication the one single chromosome, because bacteria carry a single chromosome. It's in a loop structure, no end to it, whereas eukaryotic chromosomes have two ends. They're linear. The bacterial chromosome is in a loop. That loop of DNA is replicated, and you get two daughter chromosomes that are identical to each other because they arise from the parent chromosome. And because they're identical, the cells that are produced should be identical. And when the chromosomes are replicated, or duplicated as they're sometimes referred to as, they attach to the plasma membrane. So as the cell enlarges, they're pulled along the plasma membrane away from each other towards opposite ends of the cell. And eventually the cell will divide, and two new daughter cells are produced, each containing chromosome, and the chromosome would contain all the genes that were in the parent chromosome and will help the bacterium to survive and do the things that its parent cell did. And when you have a colony of bacteria growing, if you were in my face-to-face class or lab, I could show you colonies of bacteria. If you're to sample a colony of bacteria, a colony represents a group of bacteria in the millions usually that arose from one bacterium that started dividing. So those millions of bacteria, if you were to take a sample, the sample would represent all the other bacteria in that colony, and they would all be similar, have similar characteristics, because from the one bacterium that divided many times, the daughter cells divided, you get millions and millions of bacteria that are just alike. So that would be in bacteria. And the time it takes for the bacterium to be produced and then divide and produce two daughter cells is called the generation time. Some bacteria have a generation time as little as 10 minutes, and supposedly some of the Clostridium bacterium bacteria that cause gangrene can reproduce under optimal conditions in as little as 10 minutes. And some bacteria take 15 hours or 900 minutes to reproduce. Mycobacterium, tuberculosis, Mycobacterium leprae, those are very slow growers. The bacteria that belong to that genus that cause tuberculosis, leprosy, Johne's in cattle, those are very slow growers. The mycobacteria are slow growers, so it takes months sometimes for the symptoms to show up. Tuberculosis takes about 3 months on average for the symptoms to show up once a person contracts that bacterium. And so being slow growers helps to understand why it takes long for the symptoms to arise. So there's quite a bit of difference between different bacteria. E. coli, which is one of the most studied bacteria, has an optimal generation time of 20 minutes under the best conditions. So if you grow it at body temperature, give it plenty of nutrients, it can divide about every 20 minutes. And that's one of the reasons why E. coli is a popular bacterium to use in lab conditions, because it grows quickly. Moving from the bacteria, the prokaryotic cells, to eukaryotic cells, and talking about cell division and the cell cycle. With the eukaryotic cells, scientists are very interested in the cell cycle, and this is how the cell grows, how it prepares for division, and how long that is. And this is important too in genetic manipulation, especially cloning. In the cell cycle, most of the cell cycle is made up of a phase called interphase. That's about 90% of the cell cycle. So if you look at cells through the microscope, chances are you would catch cells in the interphase portion of the cell cycle. Mitosis is just a small part, approximately 10%. The interphase is made up of 3 parts. There's G1, which is growth, and there's a significant amount of growth, production of organelles, production of enzymes, and a preparation for DNA synthesis, which happens in the S portion of interphase. And S portion of interphase, the S stands for synthesis, and that's where DNA is replicated or synthesized. The G stands for gap. It's not a gap as in nothing occurs. It's just the DNA isn't produced or replicated in G1. After the S portion of interphase, the cell enters G2, which is more growth, and it's a preparation for mitosis and cell division, which is cytokinesis. Mitosis is really the dividing of genetic material in preparation for cytokinesis. And in mitosis, the organism, the eukaryotic cell will start out as 2N, and it produces 2 daughter cells that are 2N. And 2N, when you see 2N, think of diploid. Di means 2, 2 ploidy, 2 sets of chromosomes. So in mitosis, you're going from 2N to 2N cells. The parent cell is 2N, the daughter cells, the 2 daughter cells are 2N. Just so happens in humans, the somatic cells of humans, that's the body cells, are diploid. And diploid for humans is 46 chromosomes. That would be the 22 pairs of chromosomes and then the 2 sex chromosomes for a total of 46. The 22 pairs of autosomes. Here's an illustration of the cell cycle. Most of it is interphase, and then a small portion is mitosis. There's G1, S, and G2, and then mitosis. At the end of mitosis, you have cytokinesis, and from the single cell, you end up with 2 daughter cells. This should be similar in most cases to the parent cell. It's important to remember the makeup of the chromosome. Going into the interphase, the chromosomes will be made up of 1 chromatid. After the S portion of interphase, this 1 chromatid is replicated, the chromosome is replicated to make two sister chromatids that are attached at the centromere. These sister chromatids are identical to each other. And this is what happens in the S portion of interphase. The DNA is increased because if you're making new cells and you need the genetic material to go into those cells, you want each daughter cell to have the same genetic information. So by replicating the parent chromosome and getting these two sister chromatids, eventually in mitosis, these two sister chromatids will be separated to form two new chromosomes. So a chromosome can be consisting of one chromatid, as you see here in these three examples, or it consists of two chromatids, as is shown here. But this is a chromosome, this is a chromosome, this is a chromosome, and this is a chromosome, from the illustration that I'm pointing to. Mitosis is made up of four phases. There's prophase, metaphase, anaphase, and telophase. I cover this maybe once or twice a year, and so to memorize it, to remember it, I think of the initials PMAT, PMAT. You can think of some acronym perhaps to help you remember that they're in that order and what happens. And at the end of telophase, we have cytokinesis, cell movement, or the cells moving away from each other to produce two new daughter cells. In prophase, the nuclear membrane breaks down, spindle fibers form, and the chromatin condenses to form chromosomes. So if you were to see a cell at the beginning of prophase, you wouldn't see chromosomes, you just see a grainy material. The chromatin is too small to see with the light microscope. But it condenses to form chromosomes, which you can see, and which you see more clearly in this photograph through the microscope here in this image that's being shown. In metaphase, the chromosomes align along the middle of the cell. And when you think of metaphase, it begins with an M, so does the word middle, and that's where the chromosomes are found. If I scroll down here, you can see the chromosomes in this picture are aligned along the middle or the equator of the cell. The next phase is anaphase. This is where chromosomes move away from the middle of the cell. Anaphase begins with A, and so does the word away. And that's what the chromosomes are doing. They're moving away from each other. In telophase, the chromosomes disappear and become diffused to appear as chromatin, which is grainy looking. And the spindle fibers disappear. And the spindle fibers are what help pull the chromosomes to opposite ends of the cell. And the nuclear envelope reappears. And then this is followed by cytokinesis, which is cell division. In plant cells, the cell division will occur along what is called a cell plate. In animal cells, there will be a cleavage furrow, whereby the cells divide along that cleavage furrow. So just as a recap, at the end of mitosis, you have two daughter cells that are like the parent cell in chromosome number. And this would be a good example of where an individual, an organism, is producing new cells for growth or repair. That's what mitosis is for. After fertilization, you have a zygote, and from that zygote, you have cells that reproduce and eventually end up as very specialized cells throughout the body. But that happens by many, many mitotic divisions, many mitosis events. Generally, when cells come in contact with each other, especially when they're grown in culture, in a petri dish or a flask in a lab, when they come in contact with each other, they stop growing. And that is referred to as contact inhibition. Your text, if you read after your text, it has a different name for the contact inhibition. They talk about density-dependent inhibition and anchorage dependence, but what many scientists refer to it as